या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Thank you. In Indian culture, we respect and show hospitality to the guests with all our heart. For this, I would like to request Dr. T.N. Gulprasad, Director, SVIS and Head, Department of Chemistry, to welcome our Chief Guest, Dr. G. Ramakrishnan, President of the Chromatographic Society of India and Honorary Director of SIES, Institute of Chromatography and Spectroscopic. Again, I request Dr. T.N. Gulprasad, Director, SVIS and Head, Department of Chemistry, to welcome the speaker of today's session, Dr. S. Narasimha, Founder Director of Astagiri Herbal Research Foundation.
Sir, I have shared.
A DSIR
I have shared the link of Chennai. Yeah.
I now request Dr. Kavita Sharma, Professor, Department of Chemistry and Convener, FDP 2023, to welcome Dr. Ajit Data, ex-advisor, Shimadzu Analytical India Private Limited.
I think you should go ahead, Ujjwala.
Just a minute, please.
Please go ahead, Ujjwala.
Okay, sir. Again, I request Dr. Kavita Sharma ma'am to welcome Mr. Deepak Villani, Vice President, Business Development at Ipsum Life Science. I humbly request visionary of our department, Dr. Kavita Sharma, to welcome Dr. R. Srinivas, ex-Chief Scientist and HOD of National Center for Mass Spectrometry at RICT, Hyderabad.
Ujla ma'am, your voice is not so clear. Shada sir, please take care. Technical committee, please take care.
Make it quick now, Ujjwala.
I now request Dr. Radha Mehra, Assistant Professor, Department of Chemistry, Sri Vaishnav Institute of Science, to welcome Dr. D.V. Prabhu, Adjunct Professor and former Head, Department of Chemistry, Wilson College, Mumbai. Again, I request Dr. Radha Mehra to welcome Dr. Deepak Parab, MD of Metro Home India Private Limited. Thank you, technical team, for the support. Now it's time to know more about the Faculty Development Program 2023. For this, I would like to invite Dr. Kavita Sharma, Professor, Department of Chemistry and convener, FDP 2023. Over to you, ma'am.
Thank you, Ujla. Am I audible?
Yes, ma'am. You are.
Respected Chief Guest, Dr. G. Ramakrishnan, President, Chromatographic Society of India and ex-director, SIES, Institute of Chromatography and Spectroscopy, Navi Mumbai, Maharashtra. Dr. K. Guruprasad, Director, SVIS, Sri Vaishnav Vidyapeeth Vishwavidyalaya, Indore. Eminent speakers, Dr. S. Narasimhan, Founder Director, Astgiri Herbal Research Foundation, DSIR Recognized Lab, Chennai, Dr. Ajit Datta, Ex-Advisor Shimadzu, Analytical India Private Limited, Mumbai, Mr. Deepak Velani, Vice President Business Development at Ipsum Life Science, Mumbai, Dr. R. Srinivas, ex-chief scientist and HOD of National Center for Mass Spectrometry at IICT Hyderabad, Dr. D.V. Prabhu, adjunct professor and former head, Department of Chemistry, Wilson College, Mumbai, Dr. Deepak Parab, MD and CEO, Metrom India Private Limited, Mr. Niranjan, Mr. Lenin, and Mr. Yuvraj. They are the lab managers, Metrom India Private Limited, Chennai, Dr. Santosh Dhar, Rector and Dean FDSR, Sri Vaishnav Vidyapeeth Vishwavidyalaya, Coordinator FDP, Dr. Radha Mehra, Assistant Professor, Department of Chemistry, SVIS, HOIs and HODs of all constituent institutes of Sri Vaishnav Vidyapeeth Vishwavidyalaya, and dear participants. On behalf of Department of Chemistry, Sri Vaishnav Institute of Science, Sri Vaishnav Vidyapeeth Vishwavidyalaya, Indore, I welcome you all in this faculty development program in collaboration with Chromatography Society of India on spectroscopic and chromatographic checks. I would like to share that Department of Chemistry, Sri Vaishnav Vidyapeeth Vishwavidyalaya, is organizing its second faculty development program. Thanks to Honorable Vice Chancellor Sir for allowing us and for giving his valuable time, guidance and support. Due to an urgent meeting with Kumar Mangalam, he could not join us. Also, I would like to thank Dr. G. Ramakrishnan, President, Chromatographic Society of India, for this collaboration and this cooperation. CSI is not for profit organization and trust established in the year 2000 with the sole purpose to propagate education on important areas of chromatography and spectroscopy. Chromatography and spectroscopy are the pillars of chemical analysis and is like an axle on which the chemical industries revolve. This is the reason that these topics are taught at undergraduate and postgraduate students of chemical sciences, biological sciences, and engineering also. The teachers who are teaching these subjects also would like to update themselves about these instruments so that they have sufficient practical knowledge to teach these techniques effectively to students. I am thankful to Dr. Deepak Parab, MD and CEO, Metrom India Limited, Private Limited, for his cooperation and permission for the demonstration of some techniques in this faculty development program. Chromatography is a vital tool used in many industries to determine the composition of a mixture. Chromatography is widely known for its role in scientific research and its importance in other industries like pharmaceutical, clinical trial, environmental and chemical safety, food and beverage, drug testing, forensics, petroleum, and molecular biology, etc. Spectroscopy is used in physical and analytical chemistry to detect, determine, or quantify the molecular and structural composition of a sample. Each type of molecule and atom will reflect, absorb, or emit electromagnetic radiations in its own characteristic way. Spectroscopy uses these characteristics to deduce and analyze the composition of a sample. Examples of spectroscopic applications like determining the atomic structure of a sample, monitoring dissolved oxygen content in freshwater and marine ecosystems, altering the structure of drugs to improve its effectiveness, characterization of proteins, respiratory gas analysis in hospitals, etc. So without taking much time, I would like to share about the sessions of faculty development program. We have 6 different sessions in which first day, along with inaugural session, we will be learning about Introduction to Spectroscopic and Chromatographic Techniques by Dr. S. Narasimhan, Founder Director of Astgiri Herbal Research Foundation, a DSIR recognized lab, Chennai. On the second day, we will get to know about GC instrumentation and applications by Dr. Ajit Datar, ex-advisor Shimadzu Analytical India Private Limited. On third day, Dr. Deepak Velani, Vice President Business Development at Epsom Life Science, will be speaking on HPLC instrumentation and applications. On fourth day, we will be knowing about MS instrumentation and applications by Dr. R. Srinivas, ex-Chief Scientist and HOD of National Center for Mass Spectrometry at IICT Hyderabad. On fifth day, Dr. D.V. Prabhu, Adjunct Professor and former Head, Department of Chemistry, Wilson College, Mumbai, will be speaking on NMR introduction and applications. This session will be followed by the presentation and demonstration of ion chromatography and Raman instruments by Dr. Deepak Parab, MD and CEO, Metrom India Private Limited, Mr. Niranjan, Product Manager for Ion Chromatography, Mr. Lenin, Product Manager for Spectroscopy, and Mr. Yuvraj, Application Lab Manager, Metrom India Private Limited. Once again, I am thankful to all the eminent speakers for accepting our invitation. I am sure this faculty development program will be highly beneficial for you all. Happy learning. Thank you. Over to you, Ujla ma'am.
Thank you, ma'am. It is my great pleasure to invite Dr. K.N. Gurprasad, Director, SDIS and Head, Department of Chemistry, for the welcome address. Please, sir.
Thank you, Dr. Roji. Chief guest for this FDP, Dr. G. Ramakrishnan. May I request others to please the mic. May I please request participants to put off the mic?
याद रखिए आप सभी माताओं बहनों से भक्तजनों से निवेदन है
Dr. Parimita, please put off the
बाबा की
mic.
इस विशाल भजन संध्या में
Sir, you are muted. Please unmute yourself. Guru sir, you are muted.
You are
Please
on mute,
unmute.
doctor. You are on mute.
Hi. On behalf of the university, Sri Vaishnav Vidyapit Vishwavidyalaya, I welcome the chief guest on this occasion, Dr. G. Ramakrishnan, our distinguished guest, Dr. D.V. Prabhu, and speakers in this FDP, Professor Kavita Sharma, Dr. Radha Mehra, Dr. Rujula Daswani, and participants. Chromatographic technique is one of the oldest technique, but one of the most important techniques which has undergone several modifications over the years and continues to be one of the primary tools in research, both in chemistry and as well as in biology. This is one technique that was initially started in biology and not in chemistry, because if we trace the history of this technique, it was first time introduced in 1900 by the Russian scientist Mikhail Svet, who used this technique to separate the plant pigments initially. And that is how it got the name also, because these pigments were colored, and the technique was given the name chromatography, so that these colors could be separated by using solvents. But it underwent a kind of a advance. A great advance was made when the chromatography became the partition chromatography, and that was somewhere between 1900 and 1950. A lot of techniques went into chromatography, and Nobel Prize in Chemistry was also given to this technique in 1952 to Martin and French for partition chromatography, which became the basis for developing all the modern techniques, starting from paper chromatography to TLC to HPLC to GC and all other techniques. In this faculty development program, we will be having experts in various fields, both from academics and as well as industry, who are well versed with these techniques, and also who know about the modern instrumentation which have been developed based on these techniques. And I am sure it will be extremely useful for all the participants to listen to these experts and learn a lot about chromatography. I welcome you all once again to FDP. Thank you. Over to you, Dr. Rachel.
Thank you, sir, for your earnest welcome address. I'm reminded of a quote by Bell Hooks. A good teacher is someone who can help you get back to the teacher within. With this saying, I would like to introduce the epitome who inspires not only to his students, but to everyone who wishes to learn, as our chief guest, Dr. G. Ramakrishnan. Dr. G. Ramakrishnan is one of the key founder members of Chromatographic Society of India, which was established in the year 2000 with the sole purpose of promoting education in chromatographic techniques and mass spectrometry. Presently, he is the president of CSI. He also helped to establish the SIES Institute of Chromatography and Spectroscopy at the end of 2013 to provide practical training on chromatographic and spectroscopic techniques to students, teachers, and industry chemists. He served SIES ICS as honorary director from its inception till 2018. He held the position of president in Indian Analytical Instrument Association from 2006 to 2009. He has occupied senior positions in the analytical industry, such as Vice President of Agilent Technologies India I'm the Managing Director of Thermo Fisher Scientific India. He has worked for several years as the Business Manager Asia Pacific of Analytical Products Group of Hewlett Packard in Singapore. Dr. Ramakrishnan did his MSc in Organic Chemistry from Mumbai University and PhD in Natural Products from Chemistry from Bath. He worked as a postdoctoral research fellow in US at Florida State University with Professor Werner Hurst and at Brown University with Professor David Kane. Dr. Ramakrishnan is much involved in delivering talks at seminars and symposia all over India on topics of chromatography and spectroscopy. He is known for his lecture on analytical instruments touching your life, especially to college students. He has received several awards and recognitions, including the Lifetime Achievement Award from Indian Society of Analytical Sciences. We rarely get this kind of opportunity to listen to the words of great personalities like him. So let us have the privilege of listening to him. It is my honor to request Dr. G. Ramakrishnan, sir, to address the audience with your motivation words. Please, sir.
Dr. K.N. Guruprasad, Dr. Kavita Sharma, convener of the Faculty Development Program, Dr. Daswani, who introduced me right now, distinguished speakers of this course, all the teacher participants of FDP, ladies and gentlemen, very good morning to all of you. I'm very pleased to be part of this program, and I'm very much indebted to the organizers in considering me to the chief guest of this, the inaugural function. I'm very excited that SVVV is arranging this FDP program, and I consider that such programs are very fundamental in the continuing efforts in building capabilities of our teachers that will immensely benefit the students. Dr. Guruprasad gave a very nice introduction to the importance of chromatography and spectroscopy, and I can only stress more on that, how we have to really work in this field, to continue to work in this field. A lot of work has been done on this. Lots of Nobel Prizes have been won on this chromatography and spectroscopy, and that will continue. The developments will continue, and it is important that we keep abreast with all the developments, otherwise we'll be left behind. And nobody would be blamed for that except ourselves. If you are left behind in this area, we cannot blame anybody. We have to blame ourselves. I remember that a few years back, there was a very good 2 or 3 days program organized by SVVV in Indore, and I was very happy to be a part of that. And we collaborated very well at that time with the university and made sure that this program went extremely well. We had also at that time very good eminent speakers, including 1 or 2 speakers from today. I consider that this FDP program is very important, and it is focusing on spectroscopic and chromatographic techniques. And I'm a strong believer that these techniques are the backbone of the chemical industry and education. In fact, I have made a beautiful slide once in my presentation that I made chromatography and spectroscopy as an axis of the chemical industry. And every industry revolves around that axis. We can consider that. It is a pivotal, it is the backbone. Chromatography and spectroscopy is the backbone today of the chemical analysis. And Dr. Kavita Sharma did impress on that, saying about the backbone of this industry. Any industry you take, such as environmental where it considers the analysis of air, water, soil, or any of those things, pesticide analysis. Petroleum and petrochemicals, a very vast industry, and petrochemicals. Pharmaceutical, food and flavors, fragrances, forensic sciences, medical sciences, basic research and development, etc. They all depend heavily on the chromatography and spectroscopic techniques. It's impossible even to think doing anything without the application of chromatography and spectroscopy. In fact, these techniques are applied in each and every step of development of product, and it is successfully introduced to the market till the product is introduced into the marketplace and beyond. Even the marketing department heavily depends upon the chromatography and spectroscopy when they get any complaint from any of the users, the customers. Agreed. But why should we have such a training and development program? That's an important question. Only because the above techniques are not taught in depth in the universities and colleges. Though the syllabus boldly shows that these topics have to be covered. And Dr. Prabhu will completely agree with me that there is a lot of work to be done in the area of education in these areas. The universities and colleges across India are also varied in their approaches to the education of chromatography and spectroscopy. While teachers are interested to learn these techniques in depth, unfortunately, they are not able to do as they do not have access to these instruments in their places of work. That's a serious concern. It's a matter of concern that some of the teachers teaching these techniques do not have even hands-on experience. Leave aside hands-on experience. Permit me to say, because I am very much aware of this, that some of them have not even seen the instruments working condition. I would like to stress to the teachers that the responsibility of getting such training is theirs. It rests on the teachers. And they should look around where they can get the training course. And India as a country has invested a lot in science and technology, in its universities, in its research centers, and many private industries also engaged in this. And anybody is interested to help people who are interested to learn these techniques. So the world is wide open to you, to the teachers, to learn these techniques. And I assure you, if somebody does not support you, please let the head of the departments know, or the vice chancellor know about it, or the institute heads know about it, and you will be given the facility, because it's a lot of money from the government of India. So let's make use of it. It is with this big issue in mind that as early as in the year 2000, a handful of people, including Dr. Narasimhan and myself, decided to form the Chromatographic Society of India. CSI has been very successful all these years in conducting seminars, symposia, and workshops on instrumentation independently and with collaboration with the colleges and universities all across India, and thousands of students and teachers have benefited from this. I am the witness to this. I have helped in doing this, so I can say that we can do it, and we have done it. It's only during the COVID times that we could not do face-to-face activities, but I remember that we did webinars, about 20 or 25 webinars on various techniques, and all these videos are presently available on the CSI YouTube. And thousands of people are referring to that. When I go through the records, I'm very happy that there are many students and teachers referring to the YouTube of CSI and attending to these webinars that many experts have given. And I assure you, in order to get this expertise, these people who have given the seminars have worked many years on these techniques. So you are definitely learning this from the horse's mouth. So I plead, I propose, I request all the participants and teachers to make sure that they go through the YouTube of CSI and take advantage of these videos. And with this mind, with this proposal of training the teachers and students, that is when the CSI thought that we should establish the center, and that's when we developed this SIS Institute of the Chromatography and Spectroscopy in the year 2012, and it's going on. And I left the center in 2018 for my own reasons, for my personal reasons. And during this time, I remember, and Dr. Prabhu is also witness to that out of the group present here for sure, that we have trained hundreds of teachers and thousands of students and chemists of the industry. And with the hands-on training, with GC, HPLC, GCMS, and ion chromatography, IR, UV, etc., etc. I strongly recommend, and I believe that some of the universities have done, and more of the regional universities must develop such centers and provide the help to the teachers and students to make sure that they get the hands-on training on this instrumentation. If we believe that these techniques are important, let's make the first step forward in making sure that the universities and institutes are well equipped and they are ready to serve the students and teachers. This will help us in providing a good infrastructure and we can achieve our objectives. Our objective is make sure that we are well equipped, the teachers are well equipped, so that our future generation, the students, will benefit out of that. That's our objective. I'm very pleased that CSI is able to collaborate with SVVV in this FDP. I'm glad to see that we have very good experience and distinguished teachers in this program. Dr. Narasimhan, Ajit Datar, Deepak Valani, Srinivas, Dr. Prabhu, Deepak Parab and his team. All of them are personally known to me and they are some of the best that India can offer to you. And I'm also very happy that some of the team members from Metrom are giving you some of the training programs. I'll be done in 1 minute. Dr. Daswani, I am almost to the end of my presentation. I feel bad that I could not be personally present in this teaching faculty, along with the teaching faculty this time, due to some personal reasons. I was supposed to be away this week, and I requested Dr. Kavita, and I am not able to take part. And I know she is very upset with me on this. But anyway, I'll make up for this some other time. I am once again thankful to SVVV and the organizers for inviting me to this inaugural function as chief guest, and I am pleased that CSI could support this program in several ways. I wish this FDP all the very best, and I am pleased to declare that this program is inaugurated. All the very best, and thank you all very much.
Thank you, sir. Thank you so much. As a common saying by loyal Jack Lehman, never stop learning. For when we stop learning, we stop growing. To make us learn, I would like to introduce our first speaker, Dr. S. Narasimha. Dr. S. Narasimha did his PhD in Physical Organic Chemistry from University of Madras and postdoctoral research with Professor Herbert C. Brown, which was a Nobel laureate, in Purdue University, Indiana, USA. Dr. Narasimha has worked as senior scientist in IBM, Nitro Nobel Basic Research Institute, Bangalore, from 1982 to 1988, and as Associate Director, Spec Science Foundation, Chennai, 1988 to 2003. He is the founder director of Hastagiri Herbal Research Foundation, a DSIR recognized lab at Chennai, from 2003 to present. Since 4 decades, he has gained wide exposure in many areas of chemistry, microbiology, and analytical techniques. However, he has been focusing on the chemistry of herbs and contributed to their validation. His work has 2 major impacts: in agriculture for crop protection and in natural products for the development of standardization protocols for plant products. In 1999, to appreciate their contribution, he was given the Tamil Nadu Scientist Award by the Tamil Nadu State Council for Science and Technology. Dr. Narasimha has also developed environmentally safe botanical products from neem and herbs for the control of insects and fungi. The products developed were commercialized by SPIC. He was also given the Technology Transfer Award for the same, recognizing the need for standardization and validation of herbs for their role in nutraceutical and therapeutic values. Dr. Narasimha set up the analytical facility to isolate the active components and standardize the products using chromatographic techniques. He has successfully separated active fractions from C. buckthorn, a project supported by DRDO. He has also screened approximately 50 herbs and used them as additives for soft drinks, which was sponsored by Katra Phytocare, Bangalore. In this reference, Sir has developed methods for estimating functional groups and active ingredients in food and spices. He has also brought these methods under the scope of NABL and got accreditation for reference material production to the foundation. The lab has also been approved as drug testing lab by Ministry of Ayush. Owing to his interest in utilizing the therapeutic values of herbs in food sector, Dr. Narasimha has focused on chemistry of molecules. He has also designed 300 potent molecules based on herbal chemistry and synthesized 90 molecules to cure diabetes, inflammation, and cancer. He has successfully completed several projects funded by DBT, SBIRI, and Ayush. Dr. Narasimha has mentored 40 students for their PhD in chemistry, pharmacology, biotechnology, and microbiology, affiliated to the University of Madras, Sri Ramchandra Medical College and University, and Bhaktiyar University. Over 135 publications of national and international reviews have been credited to their account. He has filed 20 patents and 8 US patents on the new chemical entities. Dr. Narasimha has served as chairman of the expert committee to provide guidelines for registration of pheromones with Central Insecticides Board, task force member in DBT, advisory committee member of many institutions, chairman of Chromatographic Society of India, currently general secretary there, through which a number of students and professionals are trained in chromatographic techniques. He has organized many workshops and conferences, mainly on crop protection and herbal technology, and also presented lectures in a number of institutions on chromatographic techniques. We are fortunate to have you with us, sir. I would like to request Dr. S. Narasimha, sir, to please enlighten the audience. Over to you, sir, please.
Please unmute Dr. Narasimhan.
Okay. Yeah. Good morning, everybody. And I thank Dr. Kavita for inviting me, and also Dr. Ramakrishnan for suggesting my name to for this seminar. And I thank the introduction person who introduced me, which is such an elaborate introduction for me. I thank her for the same. And now I think this is a very important topic for faculty improvement program, mainly because a lot of staff will have this kind of exposure and experience, which they can carry it over in their own institution for the sake of guiding the PhD students or even the students. I have been looking for a lot of, receiving a lot of students here for training and also for employment, and I find them lack in a lot of techniques, even chromatographic techniques fundamentals. I would like to start on the lecture so that I don't waste time. May I have this slide, please?
Yes,
Dr.
sir.
Kavita?
Yes, sir, just a minute.
Yeah. I have requested you to share. Yeah. So this is about Astagiri Herbal Research Foundation, which I established 20 years back, and it has got all accreditations, university affiliation, and DSIR approval, everything. And this is the overview or snapshot of our lab. Next slide. Next slide, please. Yeah. So this is the introduction to spectroscopic and chromatographic techniques. Although I thought of different topics, but then it was given to me by Dr. Kavita, and I thought this is the apt one for giving at this podium. And this also covers spectroscopic and chromatographic techniques. And this is a sort of introduction to this topic, but maybe I will be talking about my own experiences in this area. Next slide. Now, a brief about the spectroscopy and a brief about chromatography, the concepts only, not in detail, because you might have gone through or studied all these aspects in your textbooks and in your classes. Now, the spectroscopy is nothing but the relationship between matter and light, its effect on the matter. So that is the initial phenomenon that is happening place, and the interaction of electromagnetic wave with matter, the matter gives a different response based on the energy levels, X-ray, UV, IR, radio, or NMR, which is a slightly different spectroscopic technique than UV or IR. Now, what it gives is based on the nature of the material, you get absorption, and that results in a fingerprint of the particular material. It could be a complex material or a simple material or a pure compound. Still, it shows a kind of fingerprint signals. Now, the typical fingerprint obtained from the different range of wavelengths, IR, UV, NMR, NIR, NMR, and Raman, that I will be just touching on these things. And to let you know that the parameters affected are generally the material, the nature of the material, and the concentration of the material, or the kind of compounds present in that material. So these are the two parameters affected. And unique features of the material is like a chemical structure. Suppose you take a chemical, any product, any molecule that has got a unique feature, and the result of this interaction between the light and that matter gives you a fingerprint. And the response to the magnitude of the fingerprint signal is depends on the concentration. So you get these two parameters. One is the unique signal, the other one is the concentration available. Then what happens if the material is not homogeneous? And we will go through that aspect also. And this is the overall property of the matter can be studied using NIR, near infrared, and wide bore NMR or MRI. These are all the techniques which gives you the information on overall material. No need for the individual components. Now, it is difficult to decode the mixture. So that is where the chromatography comes. So now in the case of chromatography, it is a separation of components in the mixture. That is precisely what we are trying to do in chromatography. And we can blend the two, chromatography and spectroscopy, through these hyphenated techniques. Now, what are the chromatographic things which we are going to talk about is one is planar chromatography, that is thin layer chromatography. And then that has got advanced thin layer chromatography is our high performance HPTLC. And then your column chromatography, which varies with the size, material, etc. The column chromatographic, the column size and the material that are taken for separation and things like that. And then you have a flash chromatography, which is an advanced one where you can isolate molecules in bulk. And HPLC with UV characterization or photodiode array or refractive index or mass spectrum. It's a very powerful tool for identification, separation, and identification of the molecules and characterize them. And you have a preparative HPLC also to isolate the molecules, very precious molecules from nature. And all those things can be done using preparative HPLC. And what we can go through is the practical issues, problems, and developing techniques and opportunities. So that will be one area where we need to focus on so that we can understand the whole technology and then use it for our better science. See, overlapping compounds are there which need to be analyzed in a sample. And also we have to go for herbal identification and reference material production. This is generally the basic introduction that I just gave you. This, the next slide. Next slide, please. Can I go to the next slide? Please go to the next slide.
Just a minute. Next slide.
Next slide, ma'am. Yeah. Okay, this is about the basic concept of interaction of matter with the light. And you see there are different kinds of energy levels and their absorption emission spectrum that you get here. One is that X-ray, high energy, UV is lesser energy than X-ray, and then IR is lower energy. So these are all the various ranges of the wavelength which interact with the matter and give you some kind of emission spectrum. Now the matter can be homogeneous like turmeric powder. It is purely turmeric powder, which can undergo this kind of interaction with the matter, light, and you can get a signal. And the mixture is turmeric species. So therefore, this is one of the basic concepts with which the matter reacts and to the incident light. Now the next slide, you can see here the spread of the energy levels with the corresponding wavelength or the frequency, and you can see the gamma rays, X-rays, ultraviolet, and all those kind of ranges. And now this is the kind of equipment where you have the kind of and you get a signal. And in the case of UV, you get a signal as shown here. And that shows multiple absorptions, lambda max or something. And in the case of IR, it is typically fingerprint covering from 700 to 4,000 nanometers. So this is about the simple way of matter interaction. And the two very important spectroscopy are the UV and IR. Whereas IR is useful for fingerprinting of a particular compound, which can be a solid or it can be liquid. Whereas ultraviolet, generally it goes with liquid samples, liquid or solutions. And you get the characteristic response to this ultraviolet rays. Next slide. The UV techniques, this can be used for functional group analysis like proteins, carbohydrates, flavonoids, terpenoids, alkaloids, polyphenols, tannins, and their identification. And see, these are the major components in food products. And people claim a lot of nutraceutical or nutritional values. And those values, how do they determine and put them in their claims? Now, to go for analysis of this more in a validated method using ultraviolet spectroscopy, we have studied this the analysis of these various functional groups, and then included them in the NABL scope. That is, NABL is ISO 17025 2017, which is the accrediting body for the analysis of various test parameters. Now, these test parameters, none of the other labs, although they can do this, but they have not brought it into the scope of NABL. So we have brought this into the scope of NABL, measurement of these functional groups with using UV technique. Next slide. So you can see here the various kind of applications in food, UV applications in food, fruits and vegetables, and detection of chlorophyll A and B in broccoli and cabbage. Now earlier I used to see that nowadays people want moringa leaf powder, which should be quite green, retaining the chlorophyll. So therefore, how do you estimate the value of, the quantity of chlorophyll? You can use this UV spectroscopy and then do that one. And spices, you can see they are absorbing between 210 to 500 nanometers. And beverages and all these kind of things. Beverages, you use it for quality control aspects. And honey is another major product which we need to know whether it is adulterated honey or simple sugar or natural honey. And vegetable oils, other waste valorizations. So these are the what we have to observe in UV spectroscopy is the absorption maximum and multiple absorption. These are the two parameters which are necessary in UV spectroscopic studies for various compounds, various matrices and compounds. Now, why this absorption maximum? Because that will give you the best sensitivity for the functional group or the molecule. And multiple absorption is because if two molecules have got the same absorption frequency, then if one of them have got multiple absorptions, you can choose the other alternate wavelength for quantifying your product. So therefore, these are the two important parameters to note down in UV spectroscopy. So next slide. I'm not going much into, next slide, please. Into the IR techniques, which is more of matrix oriented, and you get a fingerprint, and that is very quite characteristic of many drug molecules and many other things. The main aspect of this IR technique is the cis-trans isomers, distinguishing between the cis-trans isomers, and which can be used for quantitative analysis like the Kubelka-Monk expression, which can be applied for doing some quantitative estimations in IR techniques. Next slide. Now this NMR is my favorite, and I remember now 40 years back, 1983, when I met two great associations, became two great associations for me. One is Dr. Ramakrishnan. He was at that time working for Henditron, and we were about to buy this NMR equipment in Inbri. And he came and he was giving his all features about variant FT88, which I was using already in Purdue. So I found a liking for that equipment, and Rama was instrumental in getting us this machine. Thanks, Rama, for that. And that made me get involved more into this NMR studies. Now, the other person is Dr. Manjunath, who is no more. He was working for Bruker. Though he lost his race with Rama, he was my close associate in interacting with me on all NMR theory and techniques. And through him, I learned quite a lot on the pulse programs and relaxation times and everything. Now, one of the major issues with NMR is the quantification, how to integrate and quantify the signals, and also arrive at some approximate value for the molecules. Now, especially even proton, there is some difficulty if you go for multiple scans. Single scan is probably okay. Multiple scans, you will get into the problem of integral values varying between different protons. And carbon, certainly there will be a variation because carbon nature is primary, secondary, tertiary, which are all bonded with hydrogen atoms. And their contribution to the integral value depends on the relaxation time, how fast this carbon relaxes. If it is fast, then the next pulse will give you the same quantum of energy or the to contribute towards the integration. But if the relaxation is slow, which is measured with like T1 and T2 relaxations, then the carbon nucleus is not going to give you the proper excitation. So periodic, the subsequent or continuous irradiation or giving pulses on that particular nucleus will make it saturated, and no contribution towards signal will complex. So therefore, you need to do some kind of a technique to make it relax faster, and then do quantification, quantitative carbon NMR. Now there we have used this homosphere, which is a diffuse pulse, which will diffuse all the the nucleus and get back them to the ground state. So we used this homosphere pulse. I don't know now, the technique may be advanced. It may be inbuilt right now in NMR for quantitative estimations. So that is one interesting area where we used to get this NMR, quantitative NMR for carbon also, using this homosphere pulse. And this also talks about NOE, that is no nuclear overrise and enhancement, because of the other neighboring group participation in the relaxation of the nucleus. Now this T1 and T2 study was helpful for us in determining the extent of nitration in nitrocellulose. Being an explosive company, we were making nitrated alcohols. That is, you take a polyhydroxy compounds and then go for nitration and get a polynitrates. And these compounds are very high explosives. Sometimes, like the PETN, you can use them as a primary explosive also. Now, what we have done is, because this extent of nitration is very important parameter in polymer molecules like guar gum, we tried to identify how this can be done and used this relaxation time measurements. And that was done using the Bruker Minispec instrument. And we found out that the alcohols have got shorter relaxation time due to hydrogen bonding, and they relax faster. But the nitrates were not giving that kind of faster relaxation. It takes some time. So based on these two differences, we were able to quantify the extent of nitration in guar gum. And this technique is now routinely used for non-destructive analysis of oil seeds. Now, another quantitative estimation of NMR technique, which was helpful, was when I visited Purdue again in 1992 as a visiting scientist. At that time, one of my junior colleagues was doing some kind of alkene hydroboration and then trying to do the kinetics. Dr. Brown always likes to see the kind of progress of the reaction, somewhat not quantitatively, but semi-quantitatively. He will not go into the kinetic order of reaction at all. But then he would like to see how the rate of the reaction takes place. Now, this person was asked to do that, and alkenes hydroboration, and every time some 15 minutes, 30 minutes like that, for one particular reaction, he has to take at least 5 or 6 readings, and then he has to work it up to give the alcohol and then use GC and then get the values. And that used to take him 1 week for one reaction. So now I was there and I told him, why don't you use the NMR, wherein this alkene signals you can pick it up and then go for quantifying. And then later on, I heard that he was glued to NMR machine all the time, and he did his project using that technique. Now, this QNMR as an absolute method for determining the property value of small molecules. What is the importance of this NMR is, it depends only on the proton concentration and not the molecular structure. Let it be any molecule, benzoic acid or naphthalene or anything. The proton signal is equivalent. It is quantitative equivalent. Now, if you have a reference standard, one of the molecules, it can be even benzoic acid as a reference standard, then you can use the integral values of that proton coupled with its millimoles taken, and then you can compare it with as complex a molecule like epoxy azadiradione or any natural product. Such a big molecule, you can compare the proton integrals of this with the other macromolecules and you can arrive at the purity of the compound. So this has become a major tool nowadays, the quantitative NMR study for the characterization, the property value, characterization of the property value of any new molecule using only ubiquitous reference standard like dimethyl sulfone. So this is a very interesting technique, and we have used this technique for characterizing our epoxy azadiradione, and I will come to that a little later. Next slide, please. Mass spectrum is for characterization of molecules, and what we look for here is the molecular ion, fragments, and fragmentation pattern. These are all tangibles. The molecular ion and fragments are tangible, and fragmentation pattern is what we arrive at from the various units. And why we should look in for this is because molecular ion will give you the total molecular weight of the product, compound, and fragments will give you what are all the possible fragments that you are able to get from that, and these fragments are characteristic of a molecule, and which will give you the fragmentation pattern. Now Rama is the expert in this mass spectrum, and yeah, next when he promised to give you a lecture next time, and I'm sure he will cover it up. Dispute with the regulatory authority. See, why I am saying about this fragmentation pattern is important is there was one dispute in our nearby analytical lab. They were giving a certificate for one particular pesticide or some molecule, which they found that it is not observed. So they said it is not present. But the auditor who came to see their records found that the window they have used, the time window they have used is very well within the limits of the uncertainty errors. So therefore, he argued that this molecule is present, and the lab has given that it is not present, favoring the client. So there was a show cause notice for them, and then they called me, and I went there and looked into the things. And to be helpful, I asked them what is the nature of that molecule. The two molecules were different, but having the same retention time. So therefore, this confusion has occurred. So I suggested to them that they should go for the fragmentation pattern of these two molecules, and then talk to the authorities, and they solve the problem by doing this way. The fragmentation pattern is also important in determining the nature of the molecule. And this also we found recently in pharmacokinetics in vivo issue. The parent molecule was not observed. So we found the conditions for using this technique, pharmacokinetics, using LC-MS, MS. They were using a high electron volt impact, whereas in the normal molecular weight determination, they used a soft electron ionization method. That is about 20 or 25 electron volts, and here they used 70. And under those conditions, the molecule did not survive as a parent molecule. It immediately fragmented. So I have suggested to them that they should look in for a correlation between the fragmented molecule and the parent molecule. If there is some kind of equation exists for their concentration, they could probably use it. Otherwise, they will be following only the fragmented molecule as the one which is present in the animal. So probably it may not be, but it is an artifact of the instrument. So these are some of the issues which we'll have to take care. And another one is the library match. We have to exercise very much caution in using the library match for giving the possible compounds in a particular extract or in an analysis. It's mainly because I have seen a lot of people coming up with illogical molecular structures or something for their product. And even somebody asked me to isolate that compound from the plant material, which is not present. I will show you the result in later slides. Next slide. So this is about chromatography. All these things we I just now talked to you about. What is chromatography, different techniques in chromatography, and all those things. And maybe I'm going through the same thing in the later one also. Let me see. So this is about the chromatography, as the chairperson indicated, that is a color and probably not discovered by the chemist, but by the other biologists. So need is the mother of invention. So chromatography is the technique for the separation, purification, and testing of compounds. You can see a mixture here, which gets separated through the chromatographic technique, and you get into different compounds. And it can be used for analysis, identification of the components, and purification of the components, and then quantification. Next slide. And there are a lot of chromatographic techniques and instruments available, but basically I would consider them as a planar chromatography and column chromatography. The planar chromatography is a paper chromatography, one thing, and thin layer chromatography. And column chromatography is gas liquid chromatography or gas chromatography and liquid chromatography. There are also other functional chromatographs like ion chromatograph and gel permeation chromatograph and so many other things. And it is ever expanding area of application. Next slide. Now, what is a common, next slide. The common points in this method is the analyte. Of course, these are all some of the terminologies which we use in chromatography. Maybe you know about it. Analyte, mobile phase, stationary phase, eluent, elevate, and elution. So these are all some of the terms used in the chromatographic parlance. And yeah, so these parameters we will be using in the chromatographic development of methodology or development of analytical things or preparations and all. So chromatography and TLC, that is TLC chromatography and HPTLC, the combination. Now here, as I said, the TLC can be used for identification of components, which is a very, very simple tool. And any lab can perform this one. Now, for herbal products, this TLC is used for fingerprinting of different herbs and different extracts of the same herb, so that you can find out what is the what are all the molecules that you are extracting with the different extracts. Now, HPTLC is one step further in identifying the things and giving them as a chromatogram. So like HPLC, you can get a chromatogram using HPTLC. And more to that is HPTLC can be coupled with UV. That present CAMAG instrument has got this UV attachment also to that or so. So now for each spot in the TLC plate, you can find out the UV spectrum. And that UV spectrum is probably useful if you have two isopolar molecules of differing in their chemical nature. So if you want to identify them exclusively, then you can combine that spot with UV spectrum of that spot and then make a conclusion. Now, a very interesting thing happened a couple of months back. One of the persons from my local area, he came to me and said that he bought pears, that fruit, and one was very soft and the pulp was very soft and juicy and quite sweet, whereas the other one, which is the naturally obtained fruit, was somewhat hard and not that sweet. Now he wanted me to find out why this one is very sweet. So I thought, I was not sure whether I will find out any clue for that one. But then I took that one and ran the TLC. I found that the one which was very soft and sweet had a polar spot in the TLC plate. And that was finally deciphered as ethifon. Ethifon is a plant growth regulator used by people for ripening the fruit and also for making it soft. The starch is probably hydrolyzed in this pulp. So that is why it becomes soft and more sweeter. So we could see that some of the fruits nowadays, I don't know whether it is a general practice or not, but this particular fruits had this ethypharm. So this is, we extended or rather we studied the Triphala, which is a major Ayurvedic product containing 3 fruits, Amla, Pelerica and Chebula. And we wanted to see, is there any unique molecule present in these 3 fruits, so that we can probably understand the contribution of this molecule towards therapeutic value. So you can see here the TLC was done for these 3 fruits, and then one of the formulations with a particular extract, and then gallic acid as a marker compound. You can see here how many peaks, 8 peaks were identified, and the peaks corresponding to Amla, Pelerica and Chebula are also given. And this product, the product has shown certain kind of shows the presence of all these fruits in the formulation, but at the same
Sir,
time
the
some of
slide
the
is not visible.
pardon?
The next slide is not visible.
Oh, that is the TLC plate, which is probably not showing up. Go to the next slide, please.
Yeah, here.
This one? It's visible? Okay. So here this TLC, you can see the differences between the various products or the various raw materials and products and the standard taken. And we find that there are certain unique peaks for unique spots or peaks or compounds for each product, each ingredient. So this can be a useful thing for later on developing some leads from these molecules towards developing some therapeutic action. Next slide. So how do we compare this and say that these are different is based on the UV spectrum of those parts. So you can see that RF value at 0.08, all 5 samples, they have this kind of spectrum, UV spectrum. And similarly for the 0.96 also you have similar spectrum. And so we found for the different RF values, and then finally we have given the table wherein you see the difference between the various ingredients and the compounds. Next slide. Next slide, please. Next. Yeah. Now, see, actually, thin layer chromatography, we have been using that quite extensively for fingerprinting a large number of herbal products. And also we have used it to identify the functional groups present in each track and correlated that with either tannin or protein or something present in the herb or herbal product. So that was quite useful. And later on, we wanted this column chromatography for isolation of pure compounds. This is an important thing because we were going for reference material production also, and also get accreditation from NABL as ISO 17034. We have recently obtained that accreditation, and this is one of the few labs, or maybe one of the only one lab in the whole of India for producing reference standards accredited by NABL in herbal products, herbal standards. Now here, one difficulty was, with the desacetylnimbinene, a compound from neem leaf. And this defied the isolation for months. We were trying to do this and we were not able to isolate this pure compound. So what we did was went on for a different strategy. We took a high sample size and then very low flow of mobile phase flow, and then collecting very low volumes. So that means a large number of fractions were collected. And this strategy helped us in achieving isolation of pure compounds. At least a few fractions were pure, and they were contributing to maybe more than 100 milligrams. 100 milligrams of a natural product is a very huge treasure. So without special columns for use of or use of HPLC or flash. Next slide. I'll just show here how that was done. This is the molecule, niminin. And how did we do that? Next slide. We used about 152 or 160 grams of the material extract, and then we used the stationary phase 210 grams as the for mixture, and then 363 grams, and the column bed was 560 grams. So with this,
Sir, please unmute yourself.
you'll see a pure product was obtained in this process. Next slide. So this is the way it was done with the column chromatography, and using large quantities of the sample, and then collecting various fractions and all. So and the student or the scientist had to stay overnight to effect this study. He was doing both column elution and also evaporating it at the same time. So this is what we have done. And this is one of the techniques in column chromatography. You don't have to really go for costly columns and all, costly HPLC techniques and all those stuff. Now this hyphenating spectroscopy and chromatographic techniques, which has been useful in identification, characterization, and quantification. This was hampered by the presence of other materials in the product. Now useful for as such study, these are all already I have talked about, and I don't need to say that again. Next slide. This is the standardization of quality parameters for neem oil coated urea. This was one of the issues which the Ministry of Agriculture faced. And they wanted us to, discuss with them about this issue. What is the issue is urea is given to the farmers at a subsidized rate, but the same urea is being taken up by the other people in the other industries, and they just use this for their products. So that means it is taking it at a cheaper price and then using it for a different product. So they wanted to control this, and then find out they made it mandatory that the urea should be coated with neem oil. So therefore, if you get the urea from some other place, some other industry, and check for the
Sir, please unmute.
we went in for identifying the next slide, please. Next slide. Yeah, next slide. So this urea is used for urea is used in the agriculture area and also in other places, but then it has to be coated with the neem is mandatory. So therefore, what we did is to analyze for the meliasin's content in the neem oil coated urea. So this is about neem, and a lot of applications are there for neem, and especially this neem oil. What is the problem with this neem oil is people make this neem oil by different methods. So one is directly genuine oil is obtained from the fruit dry crushing it and all, and kernel also crushing and cold press. Kernel can be extracted with solvent and you get the oil. And what sometimes people do is they just take the kernel, deep bitter it, or take off the terpenes from the kernel without even damaging the kernel. And so that gives you the desired terpenoids, and the kernel is intact morphologically. And that is sold to back into the market, and people buy that and produce the oil. But that oil is a deep bittered oil, which is not suitable for this purpose. But people make that oil also. So then what do you do? Now, if you have to identify between this type of different oils and also find out which one is really used for the neem coated urea, then you have to identify the components present in them. So this is what we did here. Next slide. Next slide, please. Next slide, next slide. This also I just now spoke. Next slide. So here you can see the different azadiractins standards here, which we have developed a method through HPLC and identified the presence of various components, and then used this for identifying the presence of any of these meliacins in the neem oil. Mostly the neem oil will contain epoxy azadiradione, and a very small amount of azadiractin. So all these things are present in the oil generally, and you can probably identify them. But in one case, we found that the nimbin is also used as one of the ingredient. And in one case, we found that the nimbin content was extraordinary, extremely high. And that was very unusual. And we were wondering about what it is. And later on, next slide. Just a minute. Here we found that the overlapping peak, it seemed to be an overlapping peak with another molecule, which is called carangin, obtained from pongamia oil or karanja oil. And now you see that the same spot, carangin, has got a multiple UV wavelengths, UV absorption, whereas the nimbin contains only absorption at 210 nanometers. So this is the deciding factor for us. And if you want to analyze, now it has also clarified that the huge concentration in the neem oil, which we analyzed, was due to the presence of carangin in that one, and which was overlapping with the nimbin. And now the proper one is nimbin. Now that means you may have to analyze this at a different wavelength to see how much of carangin is present, and then subtract it from the total peak value. Next slide. Next slide, please. So this is what we have used, and then we have solved that problem. And now comprehensive applications of spectral and chromatography in product development. So this is, next slide. The product development here, what I mean is a reference standard production. So reference marker is something like a certified reference material, which can be used for analyzing the same compound in different matrices, which will be useful or judging things for any product quality or presence of all those things. Now here we prepared this epoxy azadiradione, which has got a lot of other applications, and I'll just rush through the slides to say how it has done. Next slide. This is done according to the ISO 17034-2016 guidelines. So you need to have a production plan and control, and then next slide. Homogeneity says that means every sample or everything that we pack should be homogeneous with the integrity of the weight as well as the purity. So this assessment of homogeneity was done according to these guidelines. And next, You can see HPLC was being used, and you can see the area almost within the significant limits. Then short-term and long-term stability. Next slide. We do the short-term and long-term stability to see that this product is quite stable, and then these are also used to, done with using HPLC here. And stability at real time, that is storing it at refrigerated temperature. Next slide, please. Storing it at refrigerated temperature and following it through 1 year or 2 years and see what is the value, purity value, whether it is changing or not. You can see from the curve that it is almost stable. And next slide. So when you want to assign the property value, whether it is 98% or 97%, 96%, we need to know the other parameters also, moisture content in them, and also any inorganic impurities present. All these parameters have to be analyzed, and then you come into a property value for this product. And with all this, the regulatory authorities are not satisfied, because this is only one technique that we have used, HPLC. So they want to show the same values or approximate values using another technique. So that is where now we have used NMR. So please go through fast. Next slide. Next slide, next slide, next slide, next slide, next. This is your alternate method of assessment of, previous one. Alternate method for assessment of purity. Can you go to the previous one? Yes. So here, this is the quantitative NMR study for this product. Now, how do you do the quantitative NMR study? You use a reference standard, a certified reference standard, which is dimethyl sulfone. And you take a known weight of that one and known millimoles or molar concentration, and then run the NMR of that. It can be internal or external also, but mostly internal we do, so that we don't miss on the other conditions of NMR experiment. So then we take the compound of our interest, and that also taken in molar concentrations, and we do the NMR analysis, looking for a specific unique peak. But in this case, because the internal standard is dimethyl sulfone, there is only one peak. And for our product, there are large number of peaks, but we pick up on 1 or 2 of those signals, and then we integrate them. And after integration, we find the purity or the value, the concentration of the product in the material that was taken. So here it gave around 99.7% pure and things like that, but that is with the reference to, next slide. Next slide, ma'am. Yeah. With the reference to the different peaks, you can see the different values of purity obtained from the NMR integration, and don't neglect any of them or throw it off. And then we arrive at the percentage purity, and this is compared with the HPLC percentage purities, and then we arrive at a property value. Next slide. This is what is given here, and then it is statistically treated, and we get a around 97 or 96 plus or minus some values for the property value for the product. Now, to use all these spectroscopic and chromatographic techniques and characterize this molecule, we have done that one here, and you can see the carbon NMR, proton NMR, and then the HPLC, HPTLC, and X-ray. All these things have been taken to prove the molecular structure and also the purity of this molecule. And this is used for, next slide, characterizing or giving the proper label for this product. Next slide. And also we have the metrological traceability, so that it is now graduated as a certified reference material. Next slide. Next slide. So for this work, we were awarded the second prize from NABL in the 4th PTP RMP conclave held 3 years back. Now I talk about this last few slides. Chromatographic cues and meiotic views. I talked about this in Vellore Institute of Technology. What are the cues now? You know, the cues that we get is the retention time and integral values. And what is a meiotic view? Meiotic view is something like this. You can see. Next slide. Next slide. Next slide. Yeah. So you can see that people, you know, blind people looking at the elephant, calling it with the different, you know, properties. They are giving different properties for that same one.
Sir, please unmute.
Next slide. When people read the chromatographic clue, what are the clue here is the retention time and the nature of analyte. The area depends on the amount. So the retention time and area, when we look at this one, we tend to correlate something different. So that is what I showed you earlier about the nimbin and Karanjin, where these two are coming at the same position. The area is magnified, but then the UV has sorted out that issue. So that Q, if it had been misunderstood by any other analytical lab, they would have given a very huge value for nimbin, which is not correct. So that is the one thing which I have shown here. The next slide, the exaggerated value versus actual overlapping. That is the name story I have already told about. And the quality of neem oil, please go through fast. This is, I told you already that there are some repetitive slides here. Please pass through that. And there was a dispute in value of active compound because of lack of purity of standards from different sources. So that is okay. Now next thing is we need to quote the reference standard from where we have got. Now this is another one. Next slide. Next. This was a request I got from one person, and they wanted me to prepare this highlighted compound from Terminalia arjuna plant. This is a natural plant, but this compound is not natural. Then I was breaking my head how to find out whether this is actually present in Terminalia arjuna or not, and found out that they have only taken the mass spectrum library into consideration and that matching to this particular compound, and they asked me to prepare this. Then I told them this is not possible, this is not a natural product. So this kind of, you know, illogical identification and things are coming up because of the library match. Next slide. Next slide. Yeah, so the interpretation is not acceptable since the mass spectrum is showing a higher range. Also, the retention time for this is long compared to its polarity and molecular weight. So we need to consider the retention time, the molecular weight, and any standards, internal standards or something to identify correctly the kind of molecule present using GCMS or LCMS. So you can see in the next slide, people are asking for this compound, which is not a natural product. And they say that this is supposed to be present in the aqueous extract of Terminalia arjuna. So you can imagine how things will be. So next slide. In one of the biological extracts, after enzymatic processing, they got one extract, and they did the LCMS. And you see here, they took only this major peak as the molecule, active ingredient. But they missed out on the two other peaks. Next slide, madam. This is the LCMS of a bioproduct. The bioproduct was obtained after fermentation process, and the LCMS was taken. And in the LCMS, you can see that there is a major peak that was identified, and they just said only this molecule is the active ingredient, but they forgot to use this smaller identity of the molecules, 214 and 242. These could be, if you look at that one, 40 units difference, and it could be a carbonyl or carboxyl group, which can be another attachment or a group attached to the molecule. Next slide. So this was a major peak identified. Next slide. And if you see the structure of the compounds, you can see the different molecular weights here. And 173 was taken as the thing. But supposing you have another one added, then it could be going, it could be satisfying the other two peaks, which are so though very small. So this is one of the points which we ignore when we look into the mass spectrum. And so you need to take each and every peak into consideration before you conclude this is the actual molecule. So the acceptance of library database as true indication of the compound is a complacency in detailed study. So I don't want to go much into these, have been already talked about, and therefore I finish my talk with this and leave it to the audience for any questions. Thank you.
Thank you very much, sir, for sharing your knowledge with us. Now the session is open for the discussion. Participants are requested to please ask questions.
I had just one question to Professor.
Yes, sir.
Have you identified all the major ingredients in most of these Ayurvedic preparations?
Not in a typical product, yes, but not in all the products.
Okay.
Whatever we have taken, we have just looked in for one of the major ingredient that is likely to be a therapeutically valuable product. So that we have analyzed that and then quantified using HPLC. But then the fingerprinting we did with HPTLC, also with HPLC, but HPTLC was very easy because we can do it and cheaper also. And we did that HPTLC fingerprinting for almost 50 to 55 herbs extracts. And those extracts were used by Pepsi, I think, for putting it in their drinks.
Because most of these Ayurvedic preparations, they work in combination of chemicals, even if you identify the major chemical.
Yeah. You're right. So
Yeah.
that is why my idea is to study the total chemistry of at least a few herbal products. Like Triphala, we have done the study on Triphala. We have identified majority of the compounds in that one. And we did some bioinformatics study also with those compounds because Triphala is mainly meant for ulcer. So we tested this against ulcer and also diabetes. And we found that it is working pretty well for ulcer. On diabetes, like that acarbose or something, it was zero. It was not effective like acarbose. So these are all only the docking scores, but practically also we can see that acidity is very much controlled by Triphala. And diabetes, I'm not sure.
Because I feel that there have been less
support for marketing the product, unlike our allopathic products. So, but our foundation is interested in studying all this chemistry from different herbs, and then also simplify. Sometimes people use 10 herbs and 15, 20 herbs. Even Kabasora Kodinir, which people are using now, has got 20 herbs. Goes against COVID control. But then why all of them are required or not? So we can draw the, identify the compounds already mentioned in the literature and see whether they are actually present using some kind of this techniques, LCMS or something, and then take those molecules and go for kind of bioinformatics study on protein targets and identify what is the real herb that is important needed for this kind of control. We found, we call one particular product in my earlier work 2 years back, we did with one product, we call it Anupana, which is a vehicle for all drugs. And that one, when we did LCMS, we found hesperidin in that and epicatechin. So then we also found during our bioinformatics study that hesperidin is very important in virus control and catechin and also embolin. So we now prepared another product, which we call it Triberry. And this Triberry has got all these ingredients in an adequate dosage so that this product will go for immunity. So still I am yet to release this product in the market. But this will be based on hesperidin content, epicatechin content, and embolin content.
Thank you.
Thank you, sir.
Thank you, sir. Participants are requested to fill the feedback link. It's in the chat box. In a nutshell, today's session began by making the participants familiar about the FDP by convener of the program. Then Dr. K.N. Guruprasad, in his welcome address, mentioned about the history of chromatography and its relation with biology and chemistry. Then Chief Guest Dr. G. Ramakrishnan highly appreciated the efforts in the form of FDP 2023 and mentioned chromatography and spectroscopy forms the backbone of research labs and industry. Speaker Dr. S. Narasimha started by defining spectroscopy and explained about whiteboard techniques in addition to UV, IR, NMR, MRI. Then he switched on to explaining chromatography. Sir also emphasized on quantifying parameters with reference to vivid techniques and also mentioned about the basis of QNMR. He also explained about LCMS and MS library match and also about plant growth regulators by citing a real-life incident and reference partners. So all I can say, we have collectively dived into the ocean of knowledge. To express a word of appreciation and gratitude, we have with us Dr. Radha Mehra, Assistant Professor, Department of Chemistry, Sri Vaishnav Institute of Science.
Thank you, Dr. Ujjwala. Such a prayer that cannot be seen or touched. It must be felt by heart. I, Dr. Radha Mehra, Coordinator FDP 2023, Assistant Professor, Department of Chemistry, Sri Vaishnav Visvadhyalaya, behalf of Organizing Committee FDP 2023, Department of Chemistry, Sri Vaishnav Institute of Science, Sri Vaishnav Vidyapeeth Visvadhyalaya. First of all, I would like to give thanks to today's chief guest, Dr. G. Ramakrishnan, President, Chromatography Society of India, and Honorary Director, SIES, Institute of Chromatography and Spectroscopy, Navi Mumbai. I would like to give special thanks to invited speaker, Dr. S. Narasimha, Founder Director of Astgiri Herbal Research Foundation, for his wonderful and knowledgeable lecture. I would like to give thanks to Dr. Upinder Dhar, Vice Chancellor, Sri Vaishnav Visvadhyalaya, Indore, for his wholehearted support and encouragement. I would also like to give special thanks to Dr. K.N. Guruprasad sir for his their vision in guiding this endeavor. I would also like to give thanks to Dr. Kavita Sarma, convener of FDP, for help, support, and guidance. Finally, I would like to give thanks all chemistry department, especially Dr. Ujjwala Daswani ma'am, for her hard work and help, which has paved the way towards the successful conduction of the event. Special thanks to Dr. Anand Rajawat sir for providing us all the technical support. I take this opportunity to thank the technical staff member for providing their persistent support as when required. I would like to give thanks all faculty, students, and all of you who are attending the program. Just because of you, all these events is going successfully. Thanks to all the people who helped us either directly or indirectly to make this program happen. Thank you again and happy learning and keep learning. Have a nice day ahead.
Thank you, ma'am. I request everyone to please switch on your camera so that we can take selfies together and capture this moment forever. Thank you so much. Now I request everyone to kindly rise for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all for your cordial joining. Thank you.
Thank you, Professor Ramakrishnan.
Thank you, sir.
Thank you very much. Really appreciate it. I think it is going very well. And all the very best again.
Thank you. Thank you very much.
Thank you so much, sir.
Thanks to everyone. Thanks to Dr. Prabhu.
All the participants are requested to join us tomorrow at 2:30. Thank you. Thank you, Dr.